Okay, so our next speaker here is Meliah Dubus. She's from Front Range Community College, and she has plans to continue her studies in biology.
Can we restart it? Okay, so my name is Meliah. I studied Florrisant Formicids and Eocene to Modern Comparison with Sarah Leventhal with the CU Museum Collections Department. I would like to thank CU, SIRES, REX, and NSF for making this program possible, as well as thank Alicia, Jeffrey, Dana, and Beck, and everyone else at REX for their leadership and guidance. I would also like to thank my cohort for the amazing community and support we have built this summer. Next slide. You've seen the stories. You've seen it in the movies. You've seen it in headlines. You've seen it in emotionally gripping videos of the cute animals that are going extinct. We've seen it again and again and again. Climate change and the next mass extinction is kind of overwhelming. Next slide. Understanding how it will affect plants, animals, and entire ecosystems will help us better plan for the future. So we study the ant, and we study its future. But to study its future, we need to study its past. Ants first evolved during the early Cretaceous period, but they first really exploded during the late Miocene. We are looking at ants between those two times, at ants from the late Eocene 38 million years ago from the Florrisant fossil beds, and comparing them to modern day ants of the same lineages. Next slide. During the Eocene, the planet was more humid and warmer. The average global temperature was about 15 to 20 degrees Celsius warmer. The temperature gradient on the globe was smaller than it was today. So the climate of the poles had more similarities to the climate of the equator than they do now. This happened during the Paleocene-Eocene thermal maximum, a geologic time of global warming that many scientists use to compare it to today. It is climate change, but it's 5 times faster. That big spike that the arrow is pointing to is the Paleocene-Eocene thermal maximum, which is like really intense if you look at it in context. And ours is 5 times faster. Next slide. The Florrisant fossil beds are in Colorado, west of Colorado Springs. Here you can see the route from CU Boulder to the Florrisant Formation, which takes about 2 hours and 11 minutes. The Florrisant was formed during the late Eocene, known for its mammalian explosion. Periodic volcanic eruptions coated the valley with volcanic ash, killing organisms above and in the Florrisant lake. Those dead organisms settled on the bottom of the lake with volcanic ash and clay in layers that pressurized and made paper shale that we can pull apart and study the specimen inside today, which is a simplified version, but that's how we got the fossils. Next slide. We first took high-resolution pictures using Helicon Focus, Helicon Remote, and a stack shot setup. Then we measured the pixels using ImageJ and converted them to millimeters using a scale bar. Then we were able to use RStudio to compare the ants of the Eocene to modern day ants. When we noticed a significant difference, we looked into it further for reasoning or an answer as to why they were different. Next slide. We found that Protostega got 2.62 millimeters shorter and 0.67 millimeters thinner on average when they evolved into the modern Azteca and migrated from Colorado to Brazil, some sticking to similar ecosystems and some diversifying. Affengaster's head shape changed into 0.34 millimeters thinner and 0.22 longer head to aid them in their new microhabitat when they moved from the trees to living underground. Overall, the ants from the Florrisant fossil beds have gotten 1.21 millimeters shorter and 1.47 millimeters thinner as they've diversified from the rainforest that was there to other ecosystems like urban, deciduous, desert, etc. areas. Next slide. From the Eocene to now, ants have on average gotten smaller. The diversification of ecosystems from tropical areas has made it so being smaller is advantageous. The sprawling urbanization has also led to smaller ants. However, as urbanization continues to push ants to be smaller, climate change, which is increasing temperature and humidity, pushes ants to be larger. And there might not be a happy middle ground, and the ants might be stuck between a hard place and a hot place. Next slide. With any research, there are limitations. Comparing a fossilized ant that was crushed under a few millennia of sediment and a modern 3D ant is imperfect. Also, the measurements are imperfect as ants are not box-shaped and can be very organically shaped. So one measurement of a thorax can be very different than another. We did test the variance and all the sample measurement with a sample, and all of them were 1% or lower besides the thorax, which was 3%. In future work, I would like to be able to test the variance of size after crushing a dried out modern ant. I would also like to study more about urbanization effects on Formicidae, and of course, more time. With more time, we would be able to do more analysis, get a bigger sample size, and more meaningful data, and in general, just study the topic further. But a 9-week internship is not enough time for all of that. Next slide. Those are my references. Thank you so much for your time. If you have any questions, feel free to ask them now.
Excellent job. Excellent job, Mia. That was a very engaging presentation. As we wait for maybe some questions to come in from the audience.
A lot of messages, wow.
I wanted to mention that Mia came in very interested in entomology. I'm curious how this experience has changed or solidified kind of your interest in continuing to study entomology. You spent a lot of time this summer measuring ants.
Spent so much time measuring ants this summer. I also I loved the summer, honestly. I wanted to be an entomologist since I was like a toddler, and doing this project, I've just gotten even more interested. And before the summer, I don't know how, but I didn't like process that paleoentomology was a thing. And now I'm like, oh my gosh, I can not just study ants or bugs, I can study really old bugs. So, okay, so the first question I see is, could you tell us why urbanization leads to smaller ants? People don't tend to like bugs. So bugs in urban areas tend to be smaller because then they don't have the like wrath of humans attacking them as much. So they're out of sight, out of mind, kind of, is the main reason. Also, there is less consistently a food source because there's less plant life. You suggest that ants might be getting smaller and thinner from data from a few places. How could you dig deeper with data analysis? I think the digging deeper, I think really I would want to get a more timeline effect. So this is very comparative of the Eocene versus modern, and also just the Florrisant formation, and there's a lot more formations, but I think the biggest like dig deeper into this, I would want to get a timeline of ants and see how they have grown and shrunk and that type of thing throughout time instead of just comparing these two different times.
Excellent. And then how does the size of ants affect the species that eat ants?
I have not actually, I know that different sized ants will eat different things, but I don't actually know different species that eat ants. It was not something that I researched. I'd assume that smaller ants are eaten by smaller things, but that's not necessarily true. I know smaller ants actually do a lot of teamwork in taking down things instead of like individual fights, which is overall an ant strategy, but larger ants sometimes will be more individual in hunting. Can we make this part of an exhibit at the museum? I'm not sure they'd let it in. I do love the Ant-Man movie. It's great. Florrisant collection. Yes, they have a huge Florrisant collection in general. I didn't know about the fern specifically. I measured upwards of 200 ants, and there's still so many more.
And you do have, there's another question in the Q&A. So what criteria do you use to delineate different genera and species? Specifically, why do you classify these ants as being the same genus, even though they change in size and shape through time?
So Proto-Azteca and Azteca aren't actually the same genus. They have evolved like since then. Like Azteca is a descendant of Proto-Azteca. I didn't actually do very much taxonomy because we thought that that would be another study in and of itself, because taxonomy is very complicated. But I know that different like strategies are common. I guess I don't really know how Aphangaster has stayed one genus, honestly. It's something that I have looked into, but I haven't gotten an answer yet. But they moved from trees and now live underground, which is cool.
Okay, great. I think that's all the questions. Thank you so much, Mia.